Hey, Bio One students, Mr. Yantz. Hope you're having a good day. We're going to finish up notes, and we're going to take what we learned yesterday, and we're going to apply it a little bit here. So to wrap things up with this chapter, we're going over two different things, transcription and translation. Transcription we talked a little bit about yesterday. That's just simply RNA, messenger RNA, going and getting the code from DNA and taking it to the ribosomes. We talked a little bit about how the DNA is in the nucleus of every cell. It does not leave. So we have to find a way to get that information to the building site, which is the ribosomes, which is outside the nucleus. And messenger RNA does that. We're going to transfer the codes from DNA to RNA. It's a taxi. It's going to shuttle across there and use it. Okay. So we're going to break this down into 3 steps. Okay. And step number 1, very similar, should sound really familiar, is DNA is going to unravel. So it's going to unwind. Same enzyme as replication, helicase, is going to come up through there and break the bonds that hold DNA together. Okay, so that's the first thing that has to happen. So highlight helicase breaks the hydrogen bonds. Okay. A different enzyme this time is called RNA polymerase. It's going to go and after it unwinds, so we're exposing the bases on DNA. These little pieces of RNA are floating around, very similar to replication, except instead of having DNA nucleotides, we have RNA. The sugar's different, one base is different. AUCG, we substitute the U for the A. They're going to start finding their matches. If an A is exposed, a U pops in there. If a C is exposed, a G pops in there. So we're going to start making a daughter strand off of that DNA strand. Okay. Now, what we're forming is messenger RNA. Okay. So once we go through and we start plugging in, you can see A with U, T with A, C with G. Same thing's going up here. Okay. U and A, and same thing is going to go on the other side. What we're doing is we're just taking the information and we're copying it over to the messenger RNA. That's why messenger RNA is a single strand. It's going to take those codes and take it to the ribosomes, and we're going to talk about another process here in a second where we're going to build off of that. So really, it's very, very similar to replication. The only difference is, as you can see from this diagram, instead of DNA pieces, it's going to be RNA pieces are going to start plugging in. You can see there's ribose, different sugar, there's uracil, there's guanine. They're just going to start finding their match. So helicase is going to work their way up. We start building this daughter strand on one side. It breaks free eventually, and it's going to head to the ribosomes where we're going to use it here in just a second with translation. Okay? So hopefully that makes sense. And that's why we call it transcription. All we're doing is transcribing the information over to messenger RNA. All right? Now, translation is going to use that code, so we have to have transcription occur first. If you look at the definition here, translation is the process of building amino acids together to form proteins. We talked about yesterday how you have to have the blueprint, that's what we made with transcription, and the building materials at the same location. Okay, this is where translation, we're going to build the house. We got the building materials, we got the blueprint, we're going to put them together, and we're going to make these blood cells, skin cells, muscle cells, what have you. We're going to break this into 4 steps instead of 3. This will go in your foldable. Okay. Once that messenger RNA has arrived at the ribosomes, it literally will attach itself to the ribosomes, and it's going to serve as a pattern. The good news is we can use that pattern over and over and over again. So if I need to build 1,000 blood cells because my blood cell numbers are down, I make a messenger RNA template and I use it 1,000 times. Don't have to make it every time. Okay, so don't think we have to make a messenger RNA every time we need a cell. We can reuse those over and over and over again. messenger RNA is going to attach. Okay. And then, so let's do a little bit of this at a time. Let's look at the diagram. Okay. So that's what we're showing here. So if you look at this picture, here is messenger RNA arriving. Okay. And it's going to attach itself. This big blob thing, it looks like a bun, I think. It's already going to attach itself. So messenger RNA is in green. It's going to attach to the ribosome right here. And we're going to start making a protein. Okay. Now here's where this stuff comes in from yesterday. Okay. So let's say we're going to build an imaginary protein. Okay. So we have a second part to this puzzle. So we've got the blueprint, we got it attached, so the body knows what it's going to make. Now I got to get the building materials. So as all of us know, amino acids are built from, or proteins are built from amino acids. There's 20 of them. Okay, so remember we talked about this yesterday. There's 20 transfer RNAs. They're going to go pick up just one amino acid. They're going to go find that amino acid, grab a hold of it after we digest food, it's floating around the bloodstream, and bring it to the pattern, the messenger RNA. So the tRNA is what's going to latch on and bring those messenger RNA, bring those codes to messenger RNA. So let's do it. Now this is pretty simple. So stay with me because this gets a little confusing, but here we go. So messenger RNA has a code. And then it's going to code for an amino acid, okay? Transfer RNA has an anticodon. So we have a codon, three bases, 3 letters, and an anticodon, which are going to have to match. Let me show you. This is simple, okay? So let's say we all know that the first code is always AUG, which happens to be methionine. That's what every protein starts with, okay? Now, let's look at this. What would be an anticodon? UAC. See how that works? A with U, C with G. So UAC would match AUG. So here's how this works. So I have this transfer RNA. It's got this, on one end, it's got the amino acid. On the other end, it's got this anticodon. Okay? So it's going to go down through, let's go to this picture. It's going to go down through and it's going to find its complementary match on that pattern. Okay, so and that's why it has to be in groups of 3. Okay, so if you look right here, let's look at this. Let's blow this up a little bit. All right, so here we go. So here is what we have. I have my messenger RNA and it's got AUG. If you look really close to that picture. Here's my anticodon, the opposite codes, UAC. So here is my transfer RNA, kind of looks like a little finger sticking down here. This is the amino acid that is picked up from the food or from the bloodstream, and it finds its match and it locks in. Okay? Then the next one comes in, it locks in, and we're going to start building an amino acid chain. So let me show you. So let's just make one up. Okay? Let's say the next one is CAU, which happens to be histine. Its match would be GUA. So if you know the anticodon, you know the codon. If you know the codon, you know the anticodon. And then a bond's going to form between these 2 amino acids. Then we bring in another one. GGG, we'll make it simple. Glycine, what would be the anticodon? CCC. Get how that goes? And we're just going to keep doing that until we finally get to any one of the 3 stop codons. It can be any of these. And that tells the body, I'm done. I made a pretty short protein here. I have put everything in I need to put in. I'll put my anticodons in there so they match. Okay. We latch those in and we send that out and it becomes blood, skin, muscle, whatever. The rRNA, the ribosomes, that is basically there to make sure everything matches, all the codon messages, all the codons and anticodons match. It puts the amino acids in proper sequence, and we send it out, and it becomes whatever it needs to become. Okay. What's crazy is this: we can put 100 amino acids in this pattern. We can put 100 of them in there, which would be 300 codes, by the way. in 10 seconds. The body can do that in 10 seconds. Extremely fast. So that's pretty much what this picture was showing you. You can see, here's my first one. It locks in. The second one locks in. The third one locks in. And we just keep building. We hook them together. And then eventually that ribosome releases. And we send that protein out. And it becomes whatever it needs to be. Hopefully that makes sense. If you have questions, ask me in class. If you don't have me in class, shoot me an email if something doesn't make sense. We're going to put these diagrams and these, what I highlighted in red here, in our foldable, so we can answer some short answer questions on the test. So make sure you do that. So hopefully that makes sense. If not, ask me questions. Don't just sit there and be confused. All right? Hey, have a great day, and we will talk to you later.